Army Public School and College, Khaniwal. It's 10th class biology lecture. At chapter 14, reproduction, and its topic is artificial vegetative propagation. We have seen the natural vegetative propagation, and the vegetative propagation means to to produce offsprings from the vegetative parts of the plants, and that are the leaves, stems, and roots, other than the reproductive parts. They are known as vegetative parts. And from vegetative parts, the protrudes above the surface comes out and known as the vegetative propagation that give rise a new individual. Okay, so the artificial vegetative propagation means to say that sometimes we use these vegetative parts of the plants, we take them and we culture them, right? Hence it is known as the artificial, because it's not being natural process, it's done by humans, by the scientists, by the farmers. That is why it is called the artificial vegetative propagation. So first of all, I will tell you about the cutting. What is cutting? What happened in cutting, that you can see this is a leaf. We have cutted this leaf and we place this leaf into a new. We place this leaf into new place, you can say, right? And here it will grow. A part of stem is cut and the cut end grows into new plant when placed in moist soil. You can see that from here I cutted one leaf and then I place it on new place. I paste it here in a new place or new soil where it grows, and simply you can say cut one part of the plant from one plant and then place it in a new area where it will grow. It is known as the cutting. A part of stem is cut and the cut end grows into new plants when placed in moist soil, like mango. Yes, if you cut the part of the if you cut the leaf or any vegetative part of the mango then and grow it into the new soil, it will give a new individual. Same guava, lychee, lemon, and rose. They can we can done there the artificial vegetative by the means of cutting. Next is the grafting. What happened in the grafting? If you see in the cutting in this method, like this was the cutting in this method. Cutting may be take mainly from the stem. You can take it from the stem as we did here in this diagram, or it can be taken from the roots. The cutting must have meristematic region. You know that what are the meristematics? Meristematic tissues are those which have ability to divide. It's mean whenever we want to grow any part into a new individual, then we need to take the meristematic part. It's mean those region which has ability to divide. When cuttings are placed in suitable soil, fine. When cuttings placed in a suitable soil and under right conditions, they form roots and shoots. Roots and shoots grow and develop into plant identical to the parents. And you know that where it will grow a new plant, it will be same as the plant was here. Rose, ivory, and grapevines are propagated by stem cutting. Sweet potato is an enlarged root. Farmers place it in moist sand or soil until they produce several plantlets. Then the plantlets are removed and planted. Yes, plantlets are small leaves from them are removed and they again planted and they again make the potatoes. This process is used to produce many plants from a single plant. All new plants are exactly the same. So the artificial vegetative propagation has been very beneficial. Is it so? Is artificial vegetative propagation by means of cutting is very useful. Next is the grafting. Graft means that we have taken, this is icon, and you can see that it's one part and this is a stalk, the other part. So the stem, one part of the stem from one plant is induced into the stem of an other plant, right? We joined them. Both are different. What happens? When they are joined, they start to grow. When they start to grow, they make a new species. The stem of a plant is cut and then fitted on another strong plant and covered with grafting wax. You can see here we have covered it, right? Like it can be taken place in apples, oranges, watermelon, and ornamental plants. If you see in grafting, piece of stem is cut from one plant, right, and attached with another plant with established root system. After a while, the vascular bundles of attached stem piece and the host plants are connected each other. You can see that here the vascular bundles are present that are responsible for water and minerals conduction, and hence when these two are joined, it means their vascular bundles are also joined with each other. The stem piece and the plant begins to grow together. This method is used to propagate many roses, peach, as I already told you, trees, plum, and various seedless fruits. They can be artificially vegetative, they can be reproduced by artificial vegetative propagation through the grafting. Next is the tissue culture. What is the tissue culture technique? We know that we take a mass of cells from the body of an organism and then culture that. And we give that mass the suitable environment where it can grow. The all conditions are provided. And when it is grown into a new individual, it is called the tissue culture. A collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium is known as a compost. You can see here, this is one jar. We have taken one plant mass, like we have taken few mass or tissues of the plant, and we just put that tissues here in this medium, and we are giving it the soil, the temperature, the water, the nutrients, and the pH of the soil, each and everything that you can simply say the suitable conditions or suitable environment is provided to these tissues, and hence these tissues will grow. And when they will grow, they can make a new plant. This is called the tissue culture. As it is taking, this is being done by the biotechnological techniques. What else is there? Clone. Do you know about the human clones, class? The scientists are also making the clones. We just take the mass. Here you have seen about the plants that we took the tissues and we grow them. But in case of humans, we can also take the tissues and we will, if we grow them, then they change into the mass of multiplied mass of tissues called the callus, right? That callus is then given other conditions, the hormones, in the temperature, the hormones, the enzymes, different conditions are given to them, and they can also grow into a new individual. So that is called the cloning. So one term I introduced you here about the calluses. Calluses is the masses of tissues which is then cultured again to make a new individual. Okay, next is advantages and disadvantages of natural vegetative propagation. We have done with the artificial vegetative propagation. Now we are going to see the advantages and disadvantages. Advantages includes few points here, and these are the disadvantages. So let's start with the advantages. No need external agencies, insects, wind. Yes, when we discussed about the natural vegetative propagation, like we discussed about the stem, tubers, suckers, and rhizomes, forms, they all of them were freely doing their work. They don't need any external agencies, like they don't need pollination or wind for their reproduction, right? So simply they don't require any insects and weed or any external agency. So this is one advantage for them. Next thing, food is usually present in vegetative structures. Yes, whenever we discuss about the combs, they also contain the storage food. Bulb was also having the food. Same, it's mean that the vegetative structures, a rapid development of buds into daughter plants can take place. So food is always present here, and bud can change into daughter plant. New plants resembles parent plant in every way. Yes, they were identical, you can say. The plants that produced through the vegetative propagation, they were similar to their parents, mean they are identical. Involves only one parent. Yes, it is a method of asexual reproduction. A mean not, and sexual mean union of union of gametes, right? So it's mean here was only one parent involved, not two parents were there. That is why this is also one advantage of the natural vegetative propagation. What else can be its advantages? The offspring produced through vegetative propagation are genetically identical. Yes, parents similarity, right? One point. This is beneficial characteristics can be preserved. Yes, it's not mentioned, but you need to write it. These all advantages are also given in your book. You need to give them the bullets or just highlight them with point 1, 2, and 3. Okay. There is no need of any mechanism of pollination. Yes, no need of external agencies mean no need of pollination, right? It helped to increase number of plants at a rapid rate. Yes, rapid development of buds. We can have a good quantity with a rapid rate. The organs of vegetative propagation enables many plants to pass over unfavorable conditions. Yes, they can pass over unfavorable condition. When they got the favorable condition, they can survive. Except this, you can say that we have the plants bearing seedless fruits can be grown only by vegetative propagation. So these all are the advantages. Now come towards the disadvantages. Lack of dispersal mechanism may lead to overcrowding. The plants do not have the genetic variations. New plants are less varied, like they all are the identicals. It's mean they don't have the variations. This is the disadvantage. How it can be the disadvantage? Like if one species is destroyed, it means the whole species will be infected with one plant because all are the similar. So it can attack the whole crop. So new plants are less varied. New plants may be less adaptable to changes in environmental conditions. Yes, when they all are the same plants, they will remain same. They will not get the evolution. You can say that from the points of evolution, they can't show the changings because they all are the same. Variety can cause the environmental changes. So lack of dispersal mechanism, like what is meaning of pollination, may lead to overcrowding. Hence, its number can be increased a lot. Again, you can see here the advantages. Uniformity means they are identical. High yield means rapid rate development. Fast establishment. Again, this fast establishment, they are having the without pollination, they are having a high amount. Disadvantages includes limited adaptable qualities because they cannot change themselves according to their environments. Few individuals are produced from vegetative organs. Regenerated vegetative plants cannot survive continuous soil disturbance of the environment. So it was about the advantages and the disadvantages of vegetative propagation. So what is the meaning of micropropagation here? When I told you about the tissue culture, right? Tissue means we are taking small cells and we are culturing them. It's mean they are very micro. So you can say this as well, the micropropagation, because it uses only a small part of the plant. So it was all about our today's lecture. Any question here, please?